So good afternoon, everybody, and welcome to the first class on principles of neuroscience. So this time the enrollment, I think, has reached some kind of made some kind of a record. Must have crossed 180. So a lot of students from other departments also have taken this. Um, so I have two courses in this area. One is principles of neuroscience. Other one is introduction to computational neuroscience. So all the math stuff is covered in the comp neuro course, and this course is only biology. So now, when you say biology, students from other departments might get a bit scared because bio is all about mugging a lot of facts and remembering complicated kind of Greco-Latin terminology and all that stuff. But this course is not like that because I must tell you that there are two ways of approaching any field of biology, or I can say with more confidence, neuroscience, right? When hardcore biologists approach neuroscience, they take a more traditional approach, and where you remember all the anatomy, remember all the neurochemistry and molecular biology. So it's a zillion facts, and brain looks very complicated, like any other biological system. It's particularly complicated, yes, and that's accepted. But if you look at brain like an engineer would, right, from the point of view of first principles, brain actually looks quite simple. So after spending about 2.5 decades in this area, since my background is engineering, like most of you, and I find that brain is actually quite simple if you look at it from principles point of view. Because engineering, that's what we do. And if you take electrical engineering or mechanical, there are certain basic equations, basic laws, right? And for example, a double engineer, will start out with Kirchhoff's laws and Maxwell equations, and everything follows from that, right? But practically whole of, and then the basic, mathematics of systems theory, like the idea of a transfer function, Fourier transform, Laplace transform. But there's a very compact body of mathematics and laws, physical laws expressed in mathematics, and everything follows from that. Whereas in biology, it's generally, kind of million details all kind of thrown together. So that's why it looks complicated. So this course is about this taking this kind of a engineering style or a physics style perspective of brain. So even though we don't use any or almost nothing, almost no maths in this course, we'll take that kind of a perspective. So this class, the lecture is called Simplifying the Brain, and if you do that, and you get a whole new way of looking at neuroscience research. So let us start with some fancy statements that people make in popular media about brain. So for example, the number of neurons in the brain are more than the number of stars in the Milky Way. So Milky Way galaxy has about 100 billion neurons, and that's 100 billion stars, and that's also the kind of numbers of neurons that people have found in the brain, in human brain. So it looks something staggering, man. There's so many neurons in the brain, but you can even also turn around and ask, so what? Right? It's like saying the number of sand particles in Marina Beach are more than the number of stars in the Milky Way galaxy. So what? I mean, what kind of comparison is that? What's the point they're trying to make? Or the number of carbon atoms in my right hand are more than the number of stars in the Milky Way galaxy. So what? I mean, it's that kind of, sometimes these statements I find quite stupid. So similarly, the brain is the most complex object in the universe. Okay, so now this is almost like a 2-year-old saying, have the greatest mom in the whole world, something like that, right? It's like, I find it quite childish because, I mean, how much of the universe have you seen? Man hasn't gone, beyond the moon. And when you say object, what do you mean by object? And how far, when you say whole universe, what do what all have you seen? Right? And even on Earth, do you know all objects on Earth to make that kind of statement? So point is, these kinds of statements are quite vacuous, right? Without much substance, and they are meant to create some kind of an awe, some kind of a unreasoned admiration of brain, right, in the minds of the readers, where you feel that you can only admire the brain. You can do like kind of puja and bhajan of the brain forever, without ever being able to understand what the hell it is about. Right, and in science, that's not our objective, right? You always want to understand. Always want to nail it down and go to the bottom of it. You don't want to sit and admire things, okay, and keep staring at it with your mouth wide open like an idiot. So this is the kind of attitude that people have, oh, to infinity and beyond. So that's more for 2-year-olds, not for somebody who has a scientific temperament. So you can wonder, and why is there so much hype? In fact, even now you see that popular media constantly carries articles about something in neuroscience. People have found some 110th gene for schizophrenia or something like that. You keep wondering, so what? So why is there so much hype? One reason is, you must remember that culturally and historically, we are living in the era of big data. So science also has become like big data and big equipment. It's a big business activity. So in this kind of a science, you have to propose mega projects and lots of equipment, lots of investment. So you need to keep showing something as an output. So it's easy to just so what is easy is to get a bunch of equipment, take some recordings, make some measurements, and keep on publishing those measurements, right? And so all the time you have something new to report, right? So and you have to report a lot of stuff, and then that'll create even more buzz, and that attract even more funding. Then you can buy even more equipment, you can produce even more measurements and more buzz. So there's a kind of a positive cycle, right, which will keep on amplifying this kind of an activity without you making any progress in terms of fundamental understanding. You can have a lot of observations, but that doesn't automatically translate into understanding. But then, isn't that how biology works? And biology has always worked,
Excuse
traditionally.
me, sir.
Yeah,
Sir, there are a lot of people who can't get in into the
Sundari, can
class.
you do something? Hello, Sundari, are you there? Oh, shucks. I don't see any requests or anything. Just 1 second, I'm be online.
Sir, it says that the call has reached maximum of 100 participants. So they're not able to get in.
Okay, Sundari, are you there?
So yes, sir. So
So
I
what do we
saw it.
do?
Anyway, I'm recording this lecture. So for
No, but from
now,
next class, can we fix this somehow?
yes, sir, for sure. We're looking into it. So just for this one, we'll give them the recording. But from next class, we'll try to fix this, sir.
Yeah, so I'm sorry about that. I didn't realize that this limitation. So we'll try to fix it for the next class. Okay. Okay. So let us go on. I'm sorry about that. So that's how biology has been working right from the beginning. The picture you're looking at on this slide is a kind of a little bit of artwork drawn by Antoine von Leeuwenhoek. This first scientist who did the earliest observations of microscopic organisms in his early microscope. So he saw these things, and he saw these things wiggling in the slide, under the microscope, and he kind of used imagination a little bit and kind of interpolated stuff and had drawn all these pictures. He called them animalcules. These are like animal-like molecules. Okay, and so they look very pretty, very ornate, quite as something from a science fiction movie. But that's about it. They say nothing about what they are, how they work, and how are they able to move, how are they able to wiggle and find their way around. Nothing. Okay, so that's biology is essentially descriptive, and that is what is quite fascinating about it. And that's what is problematic about biology. Okay, so this aspect of biology has been very nicely highlighted in a quite an insightful article by Yuri Lazebnik called "Can a Biologist Fix a Radio?" or "What I Learned While Studying Apoptosis." So in this case, it's actually a takeoff on biology. So I hope people don't get offended. This is another viewpoint, and there's some merit in it. Let's look at it. So he asked the question: how do people study a radio? How do biologists study a radio? So how do they do that? So in biology, if you want to study any new system, you have to take a lot of samples of it, right? Get a lot of copies. Suppose you want to study some kind of a cancer cells. You order those kinds of cells, lots of them, and then you perform some assays on them. So similarly, you order lots of printed circuit boards of a certain radio which you wanted to study. So perhaps it looks something like what you see in this picture. Then what do? You just observe it, make observations of it. So when I observe this, what do I see? So I see that there are these blue cylinders which are kind of placed vertically, and then there are these brown ellipsoids which are also placed vertically. Then there are these kind of black boxes which have some silvery legs, right, planted in the floor, and so on and so forth. So I see a lot of these things, and I can measure them, their thickness and diameter, number of stripes, and orientation, and it's a lot of study. I mean, it's a pretty busy place, right? Look at this picture. So there's some postdoc sets out, a bunch of postdocs set out to study these kinds of printed circuit boards and they do a very detailed analysis. Okay, and that first stage of study would be called structural analysis, and this is typically how biology works. So that takes a hell of a time because it's such a complex object. And then the second stage will be functional analysis because you want to understand how it works, right? And obviously you cannot do that in biology without first understanding structure completely. So people have done that. Let's say it takes 10 years to study the structure, right? Then you come to function. Function means what do? You connect the PCB to power lines, and so in biology you assume that all things being equal, right? If I remove something, what happens? So if you want to study a gene, what is the function of a gene? That's how you pose the question. So what do? You remove that gene, and then see what happens. Is there any difference in the output, in the appearance of the body, in the phenotype, in the function, whatever? So it's like saying, what is the function of my left pink finger or pinky, left little finger or pinky? Then how do you study that? You just remove the left pinky and then see what happens, right? And that's how biology works. So okay, so in this, by the same argument, right? You remove the objects that you see on this PCB one at a time, right? Power on the PCB and then see what happens. Maybe for most objects, since there'll be so much redundancy built into the system, as you keep on pulling out these objects one by one at a time, nothing might happen. But perhaps if you pull out some crucial wire, right, maybe the wire that carries power into the PCB, right, and it completely shuts down. I mean, the radio doesn't work anymore. There's no sound coming out. Now imagine that some smart postdoc has discovered such a wire, and that will be a huge discovery, and right, and that person might get a lot of awards and offers a faculty position in top universities and so on. But I'm not making this up. This is what the article says. It's really funny. You should read that. Right. So that's how it goes on. And this kind of functional analysis will go on for another 10-20 years. At the end of it, have we understood the radio? Not really. What have we understood? Nothing. Right. We just know the appearance. We have just scratched surface for 10 years. And then we have kind of start pulling out things for another 10 years. And made zillion observation. At the end of it, have you understood radio? So now as an engineer, if you ask yourself, what does it mean to understand a radio? Right? It is essentially this. I don't need those busy pictures. Right? First of all, for radio, you need to have the concept of an electromagnetic wave. Right? There is a message signal which rides over a carrier wave, which is an electromagnetic wave. And there is a tuner circuit, a certain circuit, which resonates with an EM wave at a given frequency, and you can vary the frequency to which this circuit resonates by turning the knob on the radio. In fact, the thing is, a lot of you guys wouldn't have even seen a radio, unfortunately, but you can still find them. I suppose, I don't know. It's even I haven't seen a radio for a while, but when I was small, they were very common. People used to use them all the time. Okay, so the tuner circuit then picks up the signal at that frequency, and that is sent to a demodulator, and then that is amplified, and then fed into and then cleaned up a little bit, and fed into a speaker. You get the sound. That's all. This is radio, right? And everything else is detailed. This is the broad architecture of a radio. These are the broad elements, key elements of a radio. Any particular realization, as you find in a given radio, which is manufactured by a given company, right, will have the same overall architecture with some differences which come about when you look at the details, the circuit implementation. But if you don't understand these high-level concepts related to the radio, it doesn't, you haven't understood the radio, right? It's just you have accumulated a lot of details about the radio. Unfortunately, I mean, although it might sound like an exaggeration, a lot of things that we say we know about brain amount to this kind of thing, detailed collection, and not this kind of thing. So which is what we need to fix, which is a kind of a perspective change we need to make right in the beginning. Otherwise, we don't know what it means to know brain. So with that kind of a belief or a perspective, and combining all things that I've read and try to understand, I've written this book a couple of years ago, and which is kind of broadly what I'll be using for this course as reading material. The PDF of it is available on the net, and most of it, I mean, I've added a couple of chapters subsequently for the sake of the hard copy, but most of it is available out there. Okay, so the next reason why the situation is like this in neuroscience is that, again, we are living in the era of big data. And big data is important because, traditionally science had three pillars. The very first pillar is actually experiment, because observational science started first. And then people started building, theory, that is, applying mathematics and all that. Even the mathematics has really leapfrogged, and, the power of math in explaining physical phenomena has really begun to be realized only with the Newtonian revolution. Before that, we had applied maths and all that, and we had, for example, traditional astronomy and all that. But that's a lot like co-fitting. Once people discovered calculus and started applying it to physical phenomena, that's when science took off. So theory and experiment came first. Okay, then computational as a separate branch of or pillar of science that came about half a century ago, right from about 1940s, 50s, 60s. So it really picked up in that period. So because of big data, right? Even neuroscience, we like to collect data in every part of the brain. So, for example, let's take the single neuron itself, because as you know, brain is a network of neurons, and neuron is a fundamental cell that constitutes the brain. That's the building block of the brain. Now, neurons are interesting because any cell is basically like a shapeless blob, right? I mean, if you take an amoeba, it's like a shapeless blob. It has cell membrane, it has some cytosol or cytoplasm. Inside that there is a nucleus. Inside that you have a nucleolus. These are the standard paraphernalia of any cell. So neuron is no different in that sense, but neuron is slightly special in the sense that it has this wires sticking out of the cell body. I mean, like any other cell, it has cell body, and you can see this kind of a dark black dot somewhere in the center of these tree-like structures. And around that cell body, you have a lot of wire sticking out. Now, you can see that even in this small set of neuronal samples, you see that the morphologies, the branching patterns of this wiring has so much diversity. So that is typical of in biology. There's diversity everywhere. There's a lot of details, a lot of rich variation. Now, people have measured all these branching patterns and the number of, for a given type of neuron, what is the typical number of the fibers, and what is the branching pattern, what is the thickness of the fibers, what is the conductance value, and name it, and they have measured and they have all this data available. And all this data is published as atlases. So, for example, this neuromorph atlas that has lots of these different cell types all kind of codified and collected. So at single neuron itself, we have so much of diversity, right? And hardly have even begun to mention it. Now, if you look at a network of neurons, which is what a nervous system is, there is even more diversity. And so people wanted to understand the complete network structure of a nervous system. That is, if I have a nervous system of n neurons, right, what is their connectivity pattern? Is the ith neuron connected to the jth neuron? Okay, that problem is a pretty ambitious problem, especially if you apply it to the human brain, because human brain has 100 billion neurons. Actually, more recent, in fact, I've seen this Nature paper which came just a few years ago, and the paper says, "Oh, one of the fundamental myths of the brain has been blasted, and what we knew about brain all along is we're totally wrong." And I was wondering, like, what the hell? What did they find? So, and then the paper says that, oh, we all along we thought that human brain had 100 billion neurons. Actually, we were wrong. It only has 84 billion neurons. Okay, so that was a big myth that was blasted. I mean, I find it's a little bit of detail. That's all. So anyway, so brain has about roughly 100 billion neurons, and so the question is of Connectome project is what is the complete graph structure? So every neuron has about 1,000 to 10,000 connections. We are looking at 10 power 15 connections. So people wanted to work out that entire graph, which is obviously quite ambitious and practically a very hard problem. So people have started with a much simpler problems, like they have taken this worm called the C. elegans, or more popularly called a nematode, right? And which has only about 300 neurons, I think 302 to be precise, and has about 7,000 connections. So this graph has been completely worked out. It was one of the first kind of success stories of Connectome. So then people have started applying, they were emboldened and got encouraged and started looking at human connectome. The human connectome in its, at a very detailed level is, like I said, is very hard to work out if the ith neuron is connected to the jth neuron. But if you look at a more gross scale connectome, that is, let us say if you divide the whole brain into a few hundred regions, you just want to know if the ith region is connected to the jth region. Each region could be having billions of neurons. So that's a much simpler problem to address. There are special neuroimaging techniques, like for example, the diffusion tensor imaging can give you that kind of information. So people are, engaged in those kinds of projects. So there's a lot of work is going on that. But you can take a step backwards, right, and then ask, first of all, why do you want to understand the connectome? Because we don't even understand the basic computational principles of neuroanatomy. That is, why are things the way they are in brain's anatomy? So, for example, human brain has two hemispheres, right? Why is it so? What is the rationale behind two hemispheres? Why not have 4 divisions, right, or 12 divisions or 17? What is special about 2? Okay, so maybe you can argue and explain that this bilaterality of the brain, the fact that it has two hemispheres, has something to do with the bilateral symmetry of the body, because brain is designed to connect to the body and control the body. So therefore, maybe the brain's symmetry, bilateral symmetry, is only reflecting body symmetry. That's a good argument. Actually, it's on the right track. But so you can ask more questions like that. Then you ask the next question, which is, why is body bilateral? What is special about it? So the thing is, when we start asking why, things start getting interesting. That's when it starts looking like a science. Otherwise, it's like voodoo black magic. It's like saying you take a dark black thread and tie it, and then put some white powder on it. That's all, and that's like nonsense, right? So you need to ask why and try to find reasonable answers. So then why is there a cortex? And right, the cortex is a sheet of neurons which covers the entire brain, right, both hemispheres. Why? So I can imagine a brain in which there are masses of neurons at various points in this 3D volume, and they're all connected by some bundles of wires. Right? Because brain is basically a masses of neurons connected by some kind of a wire. But why do we need to have this thin sheet of neurons? The sheet is only about 2 millimeters to 5 millimeters thick, and this sheet covers the entire brain. It's very strange. Why do you need that kind of a sheet? Right? Before we understand, how big is the cortex and how many folds and what are the names of all the folds of the cortex, that is not that interesting to me. I want to first understand why is it even there. So similarly, there is a big structure called cerebellum, which is at the back of the brain. Why is it outside? Because all the structures of the brain are inside the brain, inside the two hemispheres. This one thing sticks out, right? Why is it outside? What is the computational logic behind its presence outside the two hemispherical volumes? And other brain structures are all inside. So point is, there are so many fundamental questions you can ask about the logic of neuroanatomy before even asking those questions. I mean, you need to first ask, investigate, and then you may or may not get any satisfactory answers. Okay, you need to begin to ask those questions. Then in that context, as you ask those questions, to give evidence to those theories, you can look for brain data of the kind, like what they find in Connectome. But data for data's sake, doing puja of data, I can never understand that. Okay, so that is big data, and that's part of the reason why there is hype. So because of all this big data situation, we have a kind of a framework of neuroscience where there's big data and tiny theory. The theory part is very weak. There are obviously theories, but I'll tell you, there are two kinds of theories. To distinguish the two kinds of theories, let me give you the analogy of take an example from astronomy or history of astronomy. There was a time when people thought that Earth was center of the universe, at least center of the solar system, and sun, moon, everything went around the Earth. Because we thought that, people thought, the Europeans at least thought that, the Earth is a very prized and a preferred location, and so therefore the whole universe goes around it. It's just basically human ego, right? Now, whereas Copernicus said that sun is the center of the solar system, and that made the planetary orbits look much cleaner, much simpler. But from that point onwards, look at the big jump that was made when Newton came up with his equations, and started the Newtonian astronomy. So thing is, at that time, before that, we just had observational astronomy, and people had uh, planetary orbits, charts about planetary orbits, and all when described in great detail. And people would just fit trigonometric time series to model those orbits. That's all. There's no logic of why they are like that. If you want to calculate where Jupiter will be 2 months down the road, yeah, the formula will give you, but it's just a curve fitting of time series, and that's not very deeply scientific. When it will work, it has a practical value, but not deeply scientific. Whereas once Newton has come up with this inverse square law of gravitation and the laws of mechanics, and based on calculus, you could not only predict the orbits of our solar system, but practically any solar system anywhere in the universe, which was a huge jump compared to what was existed before him. That's why he had the guts to say it is a universal law of gravitation. He didn't say it's a law of gravitation for the sun or for the earth or for the moon. It's universal. You go anywhere in the universe, you'll find the same laws, right, in operation. So that kind of universality is what is characteristic of science. You don't find that very often in neuroscience. At least that emphasis on the need for universal laws. It's not very common in neuroscience. So because of that, people joke that again, it's not my joke. I have other jokes, but this is not mine. So people say that there are a lot of these epicycles. So this guy called Tycho Brahe, an astronomer from about 15th century, I guess. So this European astronomer. So this guy had like really fancy observatory, and he used to measure the planets and all that. So thing is, since he was a believer of the geocentric system, so if you assume Earth is center and look at planetary orbits, they look very complicated, because basically you have two objects going around the sun in a roughly circular fashion. Let us assume, right? And then if you look at one of those objects from other object, right, you'll have two cyclical orbits superimposed. So you get epicycles. You got some kind of cycloids. And if you have accurate data, you can fit very complicated trigonometric time series to that and get good predictions. But that looks very crude. It's not at all elegant. All right. So he used to call these cycles epicycles, cycles within cycles. Okay. Now people joke that in neuroscience also you find lots of these epicycles, where things are shown in a much more complicated fashion than what is necessary, just because we are going about it in a kind of a wrong fashion. So we find in computational neuroscience there are a lot of these models, especially since we have big computers. You can always put together lots of equations and big models, complex models, just like the epicycles of Tycho Brahe. And they're not not like the Newtonian equations. So we have a lot of these curve fitting type models, right, without going very deep into the theory or into the essence of the whole system. They look nothing like F equal to ma or F equal to g into m1 m2 by r square. These are fundamental laws applicable everywhere. But curve fitting laws don't have the same kind of universality. So that's how the situation is. So because of that, the current neuroscience is theory weak, right, and data rich. Data rich is always there because it's biology, but we are theory weak. So for example, this neuroscientist and neurophilosopher Patricia Churchland says that there are no neural theories, which is kind of bold statement. She says no good ones anyway. And Indian origin neuroscientist V.S. Ramachandran, who is in UC San Diego, he says that we are at Faraday's stage in neuroscience. In fact, I had a chance to meet him, had a long chat with him in 2019. Some of our old students, IITM students, we all went and met him in his office in UCSD. We discussed for 3 hours, and all things he says is, this is the problem with neuroscience. Right, we are at Faraday's stage in the sense, at the stage of Faraday, and if you look at history of electrical theory, right, we had the Ampere's law, Faraday's law, and all that, and it was just a bunch of independent laws, right, but only much later when James Clerk Maxwell came, he could integrate all these laws into one unified mathematical framework. then electrical engineering has arrived right at that point, or electrical theory. Now we are still at a very primitive stage, like kind of like the electrical engineering of or electrical theory of middle 19th century. So because of this kind of thinking, there are lots of vague hand-wavy theories. For example, people say that thalamus is a relay station, and even if you look at some of the older textbooks of neuroscience or neurology, you will see things like thalamus is a relay station. Relay station means, right? What is a relay station? You take signal from one point, amplify it, and send it across, send it over, just send it through, let it pass. The point is, if you don't, pretty much every part of the brain can be described as a relay station. Relay station is the silliest thing you can say about any part of the brain. Because if you don't understand what it's doing, you just say it is taking a signal and then passing it on. Then if that is the case, why have that system at all? You can just lay a continuous wire right through that structure. Why do you need the structure at all? Okay, so when you don't understand, people make these interesting statements. Similarly, I once met a doctor, who has published a lot of papers, and he was very happy with himself and quite proud of his work. And so I asked him, like, he said he was working on this brain system called basal ganglia. And he said, yeah, okay. I said, I was thrilled. And I said, what do you do and what have you discovered? He said, we think basal ganglia is a comparator. What is a comparator? You take two things and compare them and see which is greater than the other one. I mean, there's a huge circuit in the brain. Imagine what it does is tell you which is bigger, A or B. Okay, so I mean, so people are happy with. They think that's an explanation. They think that that's the correct perspective. So problem is when you don't understand, when you don't know how to formulate a mathematical theory, right, and just happy with this kind of hand-wavy theories, you think you understand brain, but you understood nothing. Just like in physics or engineering, unless you put it in math, you haven't understood anything. It's just poetry. Okay, so one more reason why things are the way they are in neuroscience is a fundamental problem of all biology in a sense, but it is quite striking in case of brain, because brain, after all, is treated as a computer. It's not a great metaphor, but I mean, brain is an information processing system. For example, if you look at other organs in the body, like pancreas or heart or liver, you don't say it's processing information. It does actually. Every cell engaged in intense cell-to-cell communication. So there is signal processing going on at level of every cell. But even if you take it as a bit of a stretch, brain definitely, everybody will agree that it's like some kind of a computer. It is processing information. So different kinds of perspectives are applicable to brain, which it's not easy to apply to other organs in the body. Now, one of the fundamental things in biology is that people would like to explain everything in terms of the smallest constituents. In case of brain, that is molecules, and cells, that is neurons. So any theory of the brain, you somehow want to explain at the level of single neurons. And unless you explain at that level, but sometimes I don't even know what that means. What does it mean to explain something in terms of single neurons? Okay, so for example, let us take engineering example. So suppose you are an aeronautical engineer and you want to study the effect of wind on an aircraft wing. All right. So imagine you take a reductionist approach and you say, I haven't understood, I cannot calculate the forces of the wind on the aircraft wing unless I simulate it at the level of single molecules. And you take supercomputers and you crunch it off and you actually simulate the wind molecules, oxygen, nitrogen, all that, right, bouncing off this wing. And I mean, it'll take a whole century to simulate this if you do that. But I mean, I don't want to exaggerate, but the kinds of things that are happening in neuroscience are sort of like this, right? Whereas what do you do as an aeronautical engineer? You don't even mention molecules anywhere, right? You just look at wind as a fluid, right? Wind is not fluid, first of all, but mathematically it is a fluid, right? And then fluid is an abstraction. Fluid is not a bunch of discrete molecules bouncing around. Fluid is a kind of an average description of lots of molecules. Right, and the equations are very rigorous, and you have these new concepts, very strange concepts like viscosity and Rayleigh's number, and and so on. And using then concepts like pressure and stress and strain, all these things show up. And using that, armed with this kind of terminology and concepts, you describe quite beautifully, accurately about what is the kind of effect that the wind has on the aircraft wing. and you can design the wing in a finite time and get the aircraft flying. So thing is, what we understand in engineering, that to describe any system, you need the right kind of concepts and the right level of abstraction. So you don't want to go much to much smaller scales than what is necessary, or not much bigger scales. Sometimes when you look at objects at a big scale, right, you assume that, for example, a planet is a point mass, which is quite a horrendous thing for a biologist. I mean, planet is such a huge thing. How can you assume it's a point mass? But for my problem, under these conditions, it's a good assumption. It's okay to assume that. So in physics engineering, we know what is the right abstraction, a correct abstraction for a given problem in a given context. And if you don't do that and take a reductionist approach, things will become horrible, quickly go out of hand. So because of this kind of insistence, because of all these problems of perspective, if you look at progress in neuroscience, you find a lot of data available at the level of single cell and molecular level. You've got even subcellular stuff, molecules and all that. But when you go to systems level, that means understanding of the brain at the scale of, let us say, centimeters or tens of centimeters. All right, things get very vague. Take any major brain structure like cerebellum or hippocampus. You will find some half a dozen camps, half a dozen famous groups in the world, right? They're proposing totally different theories and they can't agree with each other. And they keep on saying whatever they've been saying and developing their own line of thought, right? On and on for a long time without making a contact with other lines of thought, right? And so that's how things remain, right? In that state of confusion. There's hardly any consensus. So because our understanding at systems level is somewhat poor, and which is the way it is because of all the problems I just described, our understanding of lots of brain disorders, both neurological and neuropsychiatric disorders, is quite vague, quite poor, right? Like bipolar disorder or schizophrenia or Alzheimer's. I mean, you will find infinite amount of data. But if you take Alzheimer's, where in the brain is the damage, what kind of anatomical changes take place, and what are the changes in different brain volumes in entorhinal cortex, in EC, and in other hippocampal fields, and so on and so forth, or neurochemistry, changes in chemical levels, and so on and so forth. But what is Alzheimer's? Why is there memory loss in this problem? Can you explain this mathematically? Right? We can't. So that is how it is. The same thing with schizophrenia or bipolar or Parkinson's or any of the major brain disorders. And because we can't understand them the way we understand the engineering system, we can't cure them, right? For most of these diseases, we only try to manage them. We have palliatives. In fact, sometimes when you have a cure, we don't fully understand how it works, right? For example, for bipolar disorder, you give something called lithium. I mean, it's not even understood why it works. It works, so we use it. So that is how it is. It looks very glitzy from a distance, but if you take a peek under the hood, right, things are very different. So therefore, in contemporary neuroscience, look at basic neuroscience. The cup is filled to the brim. In the sense, there's lots of data. But when I say it's filled, it's not in terms of understanding, but in terms of data available. Clinical neuroscience also you have lots of data, but what we can actually deliver, the comfort, the relief we can deliver to the patient, is not a whole lot. Okay, so to fix this problem, I think there is a need to develop simplified models which embody the right kind of abstraction. So just as in physics, so for every system, you have a different kind of abstractions. If you look at electrical circuits, you have the concepts of voltage and current and power, right? And for example, if you look at current flow in a wire, you don't try to look at all the bends in the wire and wire thicknesses. I don't need all that. There's a thing called current and right, and this in measured by a single scalar variable I, and that's it. That's enough. Right. So like that, we need to understand what is the right abstraction for every brain phenomena, right? Just as the way we do in physics engineering. And if you do that, I think brain looks actually quite simple, as simple as any other engineering system. For example, it's not that engineering is all quite simple. We don't understand what is complexity. Right? It's not that only biologists know what is complexity. I mean, a Boeing 787 is quite a complex system, right? A Cray supercomputer is quite a complex system. But how are we able to build and operate such complex systems? Because we are not overawed by the details, because we determine the details, but we always have our eye on the fundamental principles. Because of that, we are able to build and operate complex systems. And I think brain is no different, right? If you understand the fundamental principles and then and look at the brain, it looks quite elegant, quite fascinating actually. So with that, I think I'll. Should we take a break, or how are things going? Because do we still have this problem of not being able to bring in everybody, right?
So, yes, sir. So that's, I think, something with the G Suite account. So I used the Smail account to only send the link, but so right now we
Okay.
can do it. Fix it.
So what I will do is, even the lecture has come to a certain point, right?
Okay.
So what I will take a break for today. We'll try to fix this problem because I'm sure there are a lot of courses which have more than 100 people in the institute. So let's try to
Yes.
find out how they're managing it. Then we'll continue on Friday. Okay.
Okay.
Yeah. So see you all on Friday, because the argument have come to a certain point. Now I'll give some examples from my lab's work.